Good morning, ma'am.
Good morning.
There seems to be a slight issue because on our timetable, it's saying that we have immunology now. So
No,
I think they're just rectifying that.
yeah, I just changed it just maybe 10 minutes before because in the morning it was showing immunology. Probably it was wrongly
Okay,
published, but then I corrected it, right?
okay,
Now it
okay. Ma'am, there
should
are
be,
students
please.
in that meeting, so I'll just go inform sir.
Yeah, please. I've just changed that. Yeah, please inform them. Hello. Good morning, everyone. I hope students are joining. There are still 5 to 7 students yet to join. Okay. Okay, so in the meanwhile, we can just start with the class and let's see if they join in between or not, right? So I really hope that you all of you enjoyed your long weekend, 4 days break, uh, and are like fresh now for the classes, right? So am I audible, students?
Ma'am, you are, but your voice is quite low.
Yes, ma'am.
Okay, just a moment. Let me increase the volume. Uh, is it better now?
Yes, ma'am.
Okay, fine. So we can last lecture we discussed about the life cycle of plant viruses, in which we discussed about the different stages. We took an example of tobacco mosaic virus in that, and we discussed the different stages which are typically in a plant viruses and they infect plant viruses and they infect plants, so how they multiply, right? So the first stage was the transmission, then the infection, then how the viruses they multiply, and then how the protein synthesis and other things they takes place. So those were the we discussed about the just plant viruses and that too in reference to the tobacco mosaic virus. So today we'll discuss about again the life cycle of viruses, how they the life cycle goes on the prokaryote and eukaryotic phase, right? So prokaryotic means how they when they infect bacteria, what kind of a life cycles are there, what kind of steps are there which takes place, and another one is a eukaryotic, that is humans and animals. When they infect humans and animals, so what are the different steps in that, right? So we'll just go through. There are 6 basic steps basically. So that in each viral replication cycles or an infection cycle or a life cycle that we call, right? So first step is always that how they bind to the host, attachment, how they are attaching to the host. So this binding, it takes place between the glycoprotein spikes which are present on the surface of the viral particles and the receptors which are present on the host cell. So there are, in one slide I've given example, there are different kinds of receptors which different kind of the viruses or different viruses they recognize and then they infect or they get entry into the host cells. The protein binding is the primary factor in determining which cells a virus can infect. So it is not that a virus can infect all kinds of a cell. No, only the receptors on the cells where the receptors are present, only those cells the viruses are going to infect. So initial phase is basically identification of the receptors and then the binding and then the movement inside. For example, slight changes in the structure of the viral glycoprotein can allow a virus that usually infect another animal such as a bird or a bat and then bind to the human cell surface proteins. So this is how they can jump from animals to humans. So saying any changes in those glycoprotein can lead to the identification of the different kind of the receptors also. So once the virus is found, the genetic material, it enters the host cell. So the one then the after the genetic material is inside the host cell, then the synthesis of the viral proteins will take place, and then the replication of the viral genome will take place. Finally, these they will assemble into a virus particle or a virion, we say, right? And then at the end, these particles will be released outside the cell for the further infection. So if the virus is enveloped, the virus just buds off the cell, taking some part of the host membrane embedded with viral protein, right, which include the glycoprotein spikes for binding to the host cell. Otherwise, the host cell they lyse, releasing the naked virus particle as such directly. So this is just a diagrammatic view which tells us about the 6 different steps that how it is attachment is taking place, then the genetic material is inserted inside the cell, then the synthesis of a different viral protein is taking place, then finally there's the assembly of a viral particles and they are being, after they are being assembled into a whole virus particle, they are being released for the further infection into the cell. So the life cycle, first we'll discuss about, the life cycle of viruses with the prokaryotic host. So bacteriophages, they have always been a good model for understanding how viruses affect the cell, how they infect, and how similar processes have been observed. These are similar. You see the life cycle of viruses. If they are infecting the humans, is similar to the infecting how they are infecting the bacteria or prokaryotes. Only one difference is that major difference that we say in bacteriophages, since they do not have a proper nucleus or there's no proper nucleus is not there. So cytoplasm, the all kind of replication takes place. Whereas in eukaryotic cells, some viruses they multiply in the nucleus and some viruses they multiply in the cytoplasm, right? So the life cycle of a bacteriophage, it has been used as a good model for understanding how viruses affect the cells they infect, since similar processes have been observed for eukaryotic viruses also, so which can cause immediate death of the cell or they can establish a latent or a chronic infection. So these 2 phases are very common. So this is how they are being divided into lytic cycle and lysogenic cycle, right? Based on their action, that whether they are going to kill the cell or whether they are multiplying with the genome of the host and they are still there in the form of a chronic infection or a latent infection. So virulent phages are basically those phages which typically lead to the death of the cell through the cell lysis, whereas temperate phages, on the other hand, they go and they bind with the host genome, host chromosomes, and they get replicated with the host genome. So until such time as they are induced to make new assembled viruses or progeny viruses. So they are there, they are not going to kill the host, they are going to stay with the host there, right? So first is the lytic cycle. So what happens during the lytic cycle of virulent phages? The bacteriophage takes over the cell, it reproduces new phages, and at the end it kills the cells or it destroys the cells. One example of this is a T-even phage. It's a good example of a well-characterized class of virulent phages. And there are 5 stages in the bacteriophage life cycle. So first stage, as we discussed, attachment. Attachment is always the first stage in the infection process in which the phage it bind with the specific bacterial surface receptors. It could be the lipopolysaccharide or certain other proteins which are present on the host surface. So most phages, they have a very narrow host range and may infect one species of a bacteria or even one strain within the species. So this unique recognition can be exploited for targeted treatment of bacterial infection via the phage therapy. So phage therapy is very common these days, and it has been it has been recommended where the bacterial resistance problems are being seen in the antibiotics. When we commonly use, number of antibiotics are being used again and again. So bacteria, they develop resistant to those kind of antibiotics. So in such cases, the phage therapy is being recommended and it is very useful. Then the second stage of infection is entry or penetration. That means they are getting access, they are entering the cell. So this occurs through in case of procurement when we talk about the contractile of the tail shape, which acts like a needle and it injects the viral genome to the cell wall and the membrane. The phage head and the remaining components, they remain outside the bacteria. So this is the first cycle, which is called the lytic cycle also. So first stage is the attachment, where you can see bacteriophage is coming and attaching the bacterial to the cell. The second stage is penetration, so in which the viral DNA, it is entering the host cell, right? The third is a biosynthesis, where the phage DNA, it replicates and phage proteins are made with the help of the host genome and the host cell machinery, basically. The fourth is the maturation. The new phage particles are assembled. They're new proper phage particles or the virions particles, they are assembled. Fifth is the lysis. The cell is lysed and the kill, or another term, it is being killed and releasing the newly made phages in the environment. So the third stage of infection is the biosynthesis of a new viral component. So what happens after it is entering the host cell? Now virus, it starts its work, right? It synthesizes the virus encoded endonucleases also to degrade the bacterial chromosome. Then it hijacks the host cells to replicate, transcribe, and translates the important viral components, capsomeres or tail fibers or viral enzymes which are required for the assembly of a new virus. Now, polymerase chains are usually expressed early in the cycle, while capsid and tail protein are expressed in the later part. So, during maturation phase, new virions are being created. Now, to liberate the free phages, the bacterial cell wall is again disrupted by the phage proteins, could be a choline or a lysozyme. Now, the final stage is release. They are being released. So, mature viruses, they burst out the host cell. That means they kill the host cell in a process called lysis, and progeny viruses are liberated into the environment to infect the new cells. Now, the second stage is a lysogenic cycle, where the lysis will not take place. That means it will not kill the host cells, rather, it will stay there with the host and multiply there along with the host. So, in lysogenic cycle, what happens? The phages, they enter the cell through the attachment and penetration. The similar kind of a mechanism is there. Then an example of this kind is a lambda phages. They multiply by the lysosomes. So during this cycle, instead of killing the host, these phages, genome, they integrate to the bacterial chromosome and they become part of the host. The integrated phage genome is called as a prophage. So a bacterial host with the prophage is also known as a lysogen. So the process in which a bacterium is infected by a temperate phages is called a lysogeny, and a cycle is called a lysogenic cycle. So it is typical of or typical characteristic of a temperate phage is basically to be latent or inactive within the cell. So as the bacterium replicates its chromosome, it also replicates the phage DNA and passes it on to the new daughter cells during the reproduction. So the presence of the phage may alter the phenotype of a bacterium also sometimes, and it can bring in extra genes. For example, toxin genes that can cause can that can enhance the bacterial virulence also. Right. So this change in the host phenotype is called lysogenic conversions or phage conversion. So when they are changing the phenotype of the bacteria which they are infecting, it is called as a lysogenic conversion or a phage conversion. So some bacteria, for example, Vibrio cholerae or Clostridium botulinum, these are less virulent in the absence of a prophage. So when the prophage is there, they are more virulent. The phages infecting these bacteria, they have a certain kind of a toxin genes in their genome, and they increase or enhance the virulence of the host, and the toxin genes these are being expressed. So for example, in Vibrio cholera, the phage encoded toxin can cause a severe diarrhea. Due to the phage, presence of a phage, a severe diarrhea can be caused. In Clostridium botulinum, the toxin can cause a paralysis in the presence of a prophage. So during lysogeny, the prophage will persist in the host chromosome until induction, which result in the excision of viral genome from the host genome. So after induction has occurred, the temperate phages can proceed through lytic cycle, and then they undergo lysogeny in a newly infected cell. So this is how they carry on. So this is the example of a lysogenic cycle. The phage, it infects the cell. The phage DNA, it became attached to the host chromosome, right? Host DNA. The cell then divides and prophage DNA is passed into the daughter cell. So it is there with the host genome. So under stressful conditions, the prophage DNA is excised from the bacterial chromosome and enters the lytic cycle. Sometimes this can enter the lytic cycle, right? But if it does not enter the lytic cycle, the phage DNA replicates and phage proteins are made, new phage particles are assembled, and at the end, the cell lysis can also take place, releasing the newly made phages. So this is how it behaves, right? So if it this was about the prokaryotic cells or a prokaryotic how the virus life cycle is in the prokaryotic cells. But when we talk about the eukaryotic cells, it's a similar. You see the process is similar, only few changes are there. Otherwise, there's a attachment, there's a penetration, there's a multiplication, there's a release. A similar process is in case of a human or animal virus. So first is the viral attachment to the host cell. So this is again the same process by which the virus they infect a host organism. Then viral genome must be it get transferred from a virus particle to the cytoplasm of the host cell. So this process begins when proteins on the outer layer of a virus they bind to the attachment factors on the host cell receptors. Attachment factors are the receptors basically. So again, there are receptors which are present on the host cell. Virus identify those receptors, and then the binding will take place, and then the penetration. So these attachment factors they concentrate virus particles on the cell surface, bringing them close to the receptors, receptors that are responsible for entry into the host cell. For example, there is a CD4 is the primary receptor for human immune deficient HIV viruses. The case में CD4 is the primary receptor. But then the further interaction takes place by chemokine receptors such as CCR5 or CXCR5, which are required for the penetration. Virus receptors can be carbohydrates, they can be proteins or glycolipids. So usually it is there the single viruses, they identify or recognize multiple cell surface receptors. So that enable virus particle to bind with very high affinity, even if individual receptor have a low affinity. So they recognize multiple number of receptors. So the presence or absence of a given receptor, it determines the virus host tropism. Tropism means whether a given virus can infect a certain type of a cell or not. So viruses continuously evolve to recognize the cell surface receptor best suited for virus entry and infection of the host cell. So they keep on evolving themselves, and new receptors they are identifying, and then they are making their entry possible through those ways. So this table basically shows the number of receptors which are present in number of the viruses, their family, and the number of receptors. So just go through this. For example, adenoviruses में CAR है, then in noroviruses में HVGA receptor है, then SARS coronavirus में ACE2, this is the receptor. So every viruses they are being identified by a different kind of receptor. So second step is the viral entry. So once they are bound to the cell receptors, surface receptors, they enter the eukaryotic cells, usually through the two main pathways. So some viruses they gain directly, they gain entry to the cells directly, they fuse or they fuse with the membrane, or they penetrating the cell membrane. But most viruses they traffic into the cell by endocytosis. For example, coronaviruses also move this way. So most often this endocytosis occurs by clathrin coated pits where cargo is shuttled to increasingly acidic organelles from early endosomes to the late endosomes to lysosomes movement. Some enters at the early endosome stage, some enter at the late endosome stage, and then the lysosomes. For example, coronavirus can undergo fusion at different stages of endocytosis. MERS coronavirus fuses with early endosomes, whereas SARS coronavirus fuses with the late endosomes. Viruses that require an even more acidic pH will not undergo fusion until they reach the lysosome. So some viruses they require the acidic pH for the fusion, so they will not bind until they reach the cytoplasm. Lysozymes basically. So lysozymes we know the acidic bath, so the pH is very low there. So these endocytic and lysosomal vesicles they have a very low pH and abundant proteases that can trigger conformational changes in a virus, and that leads to the uncoating of a virus or a fusion or a penetration into the cytoplasm. So even those viruses without low pH requirement, they can exploit endocytosis as a convenient route to rapidly cross the plasma membrane and then transit through the cytoplasm to further their site of a replication, right? So how we can check this by labeling the virus particle with certain pH sensitive dyes such as PHROD, IFL green, SAPP, ester dye that can enable the visualization of the virus entry. So that can be seen, right? So this is just a pictorial view of what we have explained right now, that pathways of virus entry. So enveloped viruses can bind to the cell surface receptors and they can directly fuse with the plasma membrane. And then virus particles can also go inside by the endocytosis with escape to the cytosol occurring either from the early endosomes or the late endosome and the lysosome. So the acidic environment and proteolytic enzymes in this compartment are required for the fusion and cytosol entry by the viruses. Then viral replication. So once it has entered the cell and it is inside the cell, then it first thing it do is the uncoating and then the replication. So as intracellular obligate pathogens, now they need to take the control of cellular proteins and organelles to assist in replication, because on their own they are not having anything. So they need to use the host machinery for the replication and the multiplication. Now, the cellular components which are required for the expression of the virus gene and replications, they are organized into the replication centers. Some viruses, they replicate in the cytosols, whereas some viruses, such as herpes viruses or influenza viruses, they navigate to the nucleus for replication. So it depends on some viruses, they can easily replicate in the cytoplasm, whereas others, they replicate in the nucleus for they move to the nucleus for replication. Then there are again various methods which are used by different viruses to release the viral genome from the virion. The complete infective form of a virus outside the host cell. So certain viruses such as rhinoviruses, they expand to form pores in the endosome through which the viral genome can escape. So they form certain small pores in the endosome from which the virus, they are very small viruses, right? We know that we cannot see them by naked eyes, so they can move outside. Influenza and other viruses, they induce fusion of virion envelope within the endosomal membrane. So they fuse, the fusion with the endosomal membrane is done, and then they are finally release the viral genome outside. The process of viral genome replication varies between DNA and RNA viruses and between viruses with positive or a negative nucleic acid polarity. So some viral genomes, they contain certain instruction for producing their own proteins, such as RNA-dependent RNA polymerase in a yellow fever virus, which is a positive sense RNA virus, and SARS-CoV-2 coronavirus is a negative sense RNA virus, right? So they have their own RNA-dependent RNA polymerase. Like mRNA, positive sense single-stranded RNA, viruses can immediately be translated by host cell ribosomes, and they do not need RNA-dependent RNA polymerase in the cell, right? RNA with double-stranded RNA or negative sense single-stranded RNA or ambisense genome, they should carry their own RNA-dependent RNA polymerase protein into the cell for the transcription to take place. Now, what kind of a genome, viral genome is there, whether it is the RNA or a DNA, different kind of a polymerases are being produced. It could be RNA dependent RNA polymerase, it could be RNA dependent DNA polymerase, or it could be DNA dependent RNA polymerase, DNA dependent RNA, number of them depending upon their what kind of a genome they are having. So these polymerases, now what is the use here? These polymerases are often the target of antiviral drugs that are being used as a therapeutics in treatment viral diseases, and these can effectively incorporate the ribonucleoside or deoxyribonucleoside analogs, which enable the detection of viral RNA or DNA synthesis using click chemistry. So viral antiviral drugs are basically the viral analogs only that we use to inhibit the replication of the viruses, right? Then again, this is these are methods of viral genome release into the host cell. For example, in the A, they are just saying that rhinoviruses, they form certain kind of a pores in the endosomes, and from that they can escape. Whereas influenza B, in the B showing the influenza and other viruses, they induce the fusion of virion envelope within the endosomal membrane, so they release the viral genome. Whereas in case of a C, the many viruses, including reoviruses, they maintain partially intact capsid in the cytosol. So that act as a home base for the further application also. Right? So there are different ways of the releasing also. Then the virus maturation and release. So after nucleic acid, genome, and other essential proteins are packaged within the capsid, final packaging is taking place, right? So which further lead to assembling of a viral one viral particle. They assemble different viral proteins in form of one virion or a viral particle or a virus particle. So that is the final steps of a viral replication occur, maturation and the release. So maturation basically refers to the final changes in the immature virion that result in an infectious virus particle. Structural capsid changes are often involved, and these can be mediated by host enzymes or virus-encoded enzymes. After this maturation step, the virus is released from a host cell by either budding or lysis that we discussed in the last slide. And certain cytopathic viruses such as influenza type A and SARS-CoV-2 viruses, they undergo fusion of virion containing vesicles with the host cell's plasma membrane, and then exocytosis will take place, or budding of a new viral particle outside the cell will take place. Now, this process involves the endocytic recycling in a secretory pathways, which are known as RAB11 pathway, right? Those cytolytic viruses, such as variola major, they cause the cell membrane to rupture, followed by the death of the host cell and the release of the new viral particles into the environment or outside the cell. Then those virus particles which are gone outside the cell, they will again try to go and enter the new cells, and the new infecting the new cells. Further, they look for the new cell for the infecting, right? Because they are now released from the cell. One cell se wo release ho gaya now, but again they'll need another cell for their own multiplication and replication. So now they will go and again look out for the other cells, and then again the same process will take place, like the attachment, and there will be movement, penetration, and the multiplication, replication, and the release finally. So this is how a viral life cycle which takes place in the prokaryotic host and eukaryotic host. So as I told you in the beginning, the prokaryotic host and the eukaryotic host, it is mostly of a similar kind. There are not much differences. In prokaryotic, two main like lytic and lysogenic pathways are followed. So lytic, as the name suggests, the killing of the cell. Lysogenic, multiplying with the host genome. So they are not killing the host, they are multiplying with the host genome. So this is how they behave. Similarly, in the human also, if the phages are very virulent, the cell lysis can take place. Otherwise, the lysogenic cycle can be followed and they can live with the host. Like so, this is how a virus enters, multiply, replicate, and then they are being released. So, role of the receptors, you know, in order to identify and to further infect host is very important. Yes, Yukta, you want to say something?
Ma'am, I had a question.
Yes, आपकी आवाज क्लियर
Ma'am,
नहीं आ
when
रही है
you were
Yukta.
explaining, ma'am,
Yukta,
am I audible?
आपकी बहुत बैकग्राउंड नॉइज है. अच्छा, बोलिए आप.
Ma'am, I wanted to ask what are Clathrin coated, which you mentioned when you were explaining viral entry.
Just a moment. Viral entry in the human Clathrin
I'll
coated
write
plates.
it.
Just you just write it. I can see it. These are basically the Clathrin. Just a moment. Clathrin coated plates. These you know, they play a very important role. This is a basically protein. Clathrin is a type of a protein and which play a role in the formation of the vesicles, right? ये वेसिकल के फॉर्मेशन में इट प्ले सम काइंड ऑफ अ रोल. क्लैथरिन पिट्स का मतलब हुआ कि उसमें इट फॉर्म सम काइंड ऑफ अ पिट्स, राइट, रीजन्स ऑफ द सेल मेम्ब्रेन. So basically some kind of a pits are being formed in the clathrin is a protein. So through those pits, the receptor mediated endocytosis will take place.
Okay.
Yes. So see, whenever we talk about life cycle of a viruses, or when we talk about how viruses infect human, plants, and animals, there's always a big question in everybody's mind when we start studying viruses, or what if human viruses infect, plant viruses start infecting human? What if the human viruses start infecting plants? Though human viruses and animal viruses, few are still there common, but not all viruses are common. So here the major the answer to all this question is basically the receptors, that viruses will not affect or infect a plant or a human or a vice versa until they'll have a certain kind of a receptors. They will recognize those receptors, there is certain kind of association will take place, and then only the infection will caused by the viruses. So this is the reason that why human viruses cannot affect the plant, and why the plant viruses cannot affect the humans due to the receptors, the cell receptors. So in the life cycle, in the beginning, I mentioned one point that the viruses identify number of receptors. So maybe some receptors to a larger extent, there's a good, they identify in a better way. कुछ receptors में उनकी जो identification is not that strong. So it can, why, how does this number of viral receptors they are identifying, they helps in their survival? Because if they cannot bind to one receptor, they look for another receptor and they try to move inside, right? So that it is increasing their chances of a movement inside the host. So this is, in one line I also explained or mentioned that they are evolving. Viruses are evolving continuously to increase the receptors identification, the range of the receptors. So they are continuously evolving so that they can identify more and more number of receptors, and they can affect or infect number of the hosts and number of the cells, right? So when we talk about the viral life cycle, the 5 stages are very important. Lytic cycle and a lysogenic cycle, what are the differences is important point. And then this, this is just the overview of a viral life cycles. If we want to study, this is just the basic course, Viro 302 for a graduate student. So if we study in detail about the life cycle of the viruses, as I discussed in one pair, it is different for RNA viruses, it is different for positive sense RNA viruses, it is different for negative sense RNA viruses, it is different for DNA viruses, it is different for a circular viruses, right? Everybody has a different mechanism and their own mechanism of a replicating or a multiplying. Few steps are common. For example, viroids, they replicate by a rolling circle mechanism. They use a different kind of a mechanism for the multiplication. So the basic steps are these. But the replication of if we study the individual viruses, RNA and DNA, double stranded, single stranded, positive sense, negative sense, so the life cycle, it becomes different for many viruses or all the viruses. It becomes, there are some steps which are different. So the here in this course we are talking about the general things. We are talking about the general transmission, we are talking about the general life cycle, or we are talking about the general multiplication or a general management of the viruses. So we are not focusing on any particular virus. We are talking in general about every virus, right? So here you might be maybe in next some courses you might study the replication cycle of a viroid, or maybe replication cycle of a double-stranded DNA virus. So that you might find different from the one that we studied here, because there are steps which are different in every kind of a replication cycle or a life cycle of a virus, right? So that's all from my side in this lecture. If there's any question, please ask your question. And in the meanwhile, I'm posting one assignment for you. I created one assignment and you guys quickly do that assignment. Just a moment. Okay, here is the assignment. Let me assign it. Okay, I have assigned it, should be visible to you. Just in one paragraph, or just draw a line in between and differentiate between lytic and a lysogenic cycle of a virus. I don't know if it's visible yet. I'm still assigning. Internet is very low. Is it visible yet? No, I guess.
No, ma'am.
The net
No,
is very
ma'am, not.
slow. Yeah, actually it is just turning round and on. The net is very slow. Just a moment. Okay, this is difference between lytic and lysogenic cycle of a virus. You'll do on your Word file and then you'll attach, right? In the meantime, I'll assign it and you can see it. But you start doing it. It's not going. I posted this question in the chat box also. So in the meanwhile, you start doing it and I guess the assignment will be published. I guess it should be visible now. Done.
Ma'am, how many points should we add?
5 points sufficient.
Okay.
Maximum five. Maximum five. You can be less than that also. Okay, done, students. Can we start with the attendance in the meanwhile?
Yes, ma'am.
Please switch on your cameras for attendance. Mm, Adhish Mishra.
Present, ma'am.
Yes, and then we have Athoiba,
Present,
Ayushi Jain,
Present, ma'am.
and others. All of you, please switch on your cameras. Yes. Devanshi Sharma, Ishita, Janvi,
Present, ma'am.
okay, Kshitij,
Present, ma'am.
Manaswani, yes, you can see,
Present, ma'am.
and then Namrita,
Present, ma'am. Present, ma'am.
Nandini,
Present, ma'am.
Nehal,
Present, ma'am.
Rupal,
Present, ma'am.
Ahiba,
Present, ma'am.
Sanjana Sharma,
Present, ma'am.
Shakti,
Present, ma'am.
Shivani, Tanvi,
Present, ma'am.
Ritesh,
Present, ma'am.
Vivek, Yukta,
Ma'am, excuse me.
yes, Devanshi.
Ma'am, I'm present. Actually, my team is hanging.
Yuvraj, all of you just make sure that you all are present because these days we are downloading the attendance Excel and we are uploading directly that Excel in the Amazon. So it is automatically taking attendance. Who are present, who are absent. So actually we are not marking attendance and it is being done automatically, right? Yuvraj?
present, ma'am.
Yeah. So all of you just upload your this assignment maybe uh, by lunchtime, I guess, right? So that I can check that. And tomorrow we don't have a class, we have a practical class on Thursday, right? So I'll see you on Thursday now, right? Okay, thank you so much, students.
Thank you, ma'am.
Thank you, ma'am.
Okay. This neck